Assalamu alaikum. On behalf of Ahsan Academy, let me welcome you to a presentation uh, epithelial tissue. Actually, in this presentation, I will discuss the related concern area. Today's discussion will include the basic concept of contact areas between the epithelial cells, the types of specialized contact that exist between the epithelial cells, structure and of the basal lamella and basement membrane, and the structure and functions of the cell surface modifications we observe in the epithelial cells. Epithelial cells are very closely packed. Still, there exists some gap between the cells, and this gap is filled up with some chemical substance to maintain the adhesion between the cells. And most of the area between the cell, about 20 nanometer gap, is maintained and is filled up with protein and the calcium. This mixture works as the adhesive glue between the cells. To achieve specialized function, and this intercellular gap shows some specialized type of contact areas, especially to prevent the leakage between the cells. The gap between the cells is reduced and the plasma membrane is fused together in some areas. This is called the tight junction or jonula occludens. The strong adhesion is required in some areas, so the glue substance, that is the proteins, some fibers, runs between the membrane and keep them tightly together. This type of junctional area is known as adhesive junction or jonula adherence. Third type, there is some protein structure that passes between the cells and keep a gap like tubes inside the tube, that is the lumen. So this type of adhesion creates a cytoplasmic continuity so that information from one cell cytoplasm quickly reaches the other cell cytoplasm of the adjacent cells. This type of junction is known as gap junction, is jonula adherence. In the figure, sequentially tried to show the three types of spliced junction between the epithelial cell that exist to achieve specialized function. Suppose these are two cells. The dotted area showing the plasma membrane is a bilayer membrane, and two cells are fused together. The outer membrane is fused together. Now the two cell has three membranes. So one is supposed to have two layers, but now one layer is fused. These are the two another cell type. So this between this gap is maintained, but the gap is filled up with some fibrous substances, protein filaments, and the two cells are adhered together, kept in position by this thread-like structure. The third one is a tubular protein-made structure that is passing between the cells. So a lumen exists that makes a continuity between the cytoplasm of the adjacent cells. So the upper one prevents leakage between the cell. The second one allows the leakage, but cells are kept together by an anchoring fibers. The third one is allowing the passage of chemical substances from one cell to another. Let us discuss the junction one by one. These are the specialized junction. First is the tight junction or jonula occludens. You can see in the figure the two cell membranes are separated, but in some areas they are in direct contact and the contact line is not straight. It is zigzag in nature. This contact area encircles the total cell apex, and the outer membrane are fused together to make one. This line of fusion, that is intercellular gap, is absent, and there is anastomotic line of fusions. Tight junction is observed in the transitional epithelium close to its apical end of simple columnar epithelium. So you can see in the photographs the apical region of the cell shows the tight junction. The function, it prevents leakage between the cells and produce a barrier between the luminal fluid and the luminal structure and the part below the epithelium. This also achieves some degree adhesion between the cells and prevents separation of the apical region of the cell. Gap junction is the second type of junction. Let me discuss. It is also called zonular communicans, as it ensures the communication between adjacent cells. The intercellular gap is reduced here in this region. The hexagonal beads protein molecule traverse between the membrane. You can see in the figure the hexagonal proteins are arranged such a way that if needed, this can close the lumen or it can open the lumen, as shown in the green circular diagram. The protein channel communicates or ensures the cytoplasmic continuity through this channel, and it is not a fixed one, it is selective. The gap junction is present in the cardiac and smooth muscle, and its function is rapid transmission of the signal from one cell to another. Because of this, cardiac muscle can transmit the impulses rapidly from one place to another. Adhesive junction or zonular adherence. The intercellular gap is filled up with dense protein molecule, and this is in electron microscope become darker. The cytoplasm close to this area is condensed, and this area is known as plaque. This numerous cytoplasmic filaments are observed in this plaque area, as shown in the right upper diagram. In this way, the filaments passes through the membrane and crosses the other membrane. In this way, this microfilament anchors the part, and this forms an encircling belt near the apex of the columnar epithelium or other epitheliums. You can see three type of junction here. This one is zonular adherence. This is in electron microscope, it looks like this one. This achieves the adhesion between the adjacent cells. Desmosome or macular adherence. This is patchy, very close to the adhesive joint. It's a dense plaque is formed on the membrane side, and the intercellular area between this cell membrane is a bit dilated and filled up with the protein molecule as before, and the anchoring filament crosses these areas. These are patches, very strong adhesions. These patches are known as desmosome. Now you can see this is the tight junction, this is the adhesive junction, this one is the desmosome. Now desmosome may present near the basement membrane. One side has desmosome, other is the basement membrane. So cytoplasm is present in one side, and this type of junction is known as hemidesmosome because it looks like desmosome but is having only one side. It anchors the cell basal part with the basal lamella. Its function is to provide strong anchorage effect uh, with the basal lamella or basement membrane. As shown, let us discuss the structure of the. You see, epithelial cell rests on a non-cellular membrane. This is epithelium, and this is connective tissue deep to it, and in between there is a very thin sheet shown by the arrow, and it is non-cellular sheet on which the cell rests, and it is very thin. Electron microscope can reveal the structure of this basal lamella. This you can in the normal laboratory microscope we can see a line like this one is the junction between the plasma this in the connective tissue and the basal cells of the epithelium. If it is single layer cell, all the cell rests on the basal lamella. If it is multi-layer, then basal cell lies on the basal lamella. You can see a three-dimensional photograph on the left side. It lies between the epithelium and connective tissue and around some specialized cells like around the muscle cells, around the adipose cells, and Schwann cell. This is present in the peripheral part of the nervous system. So all connective tissue is separated from the overlying epithelium by this type of lamella, and each of these cells are covered by this non-cellular shell as mentioned in this slide. In the figure, the basal lamella is visible. The square area is enlarged to show the structure of the basal lamella. The superficial part is shown in the bracket is the lamella lucida. Lucida means pale color. It is by electron microscope we observe this as a pale area or layer. The deeper to is relatively darker and full of fine filaments. That's why it is known as lamella densa, electron dense. So this is the structure of a basal lamella. Superficial is the lamella lucida and deeper part is lamella dense. This basement membrane consists of similar two layers of basal lamella like it is shown here. If it comprises of, this is the basal lamella. So basal lamella comprises of lamella lucida and lamella densa. Another basal lamella when fixed with is similarly observed in epithelium in one side, another side epithelium is there. So in opposite direction the same thing is there. From one side we'll get lamella lucida, the densa of one epithelium, then densa and lamella lucida of other epithelium. So this become very thick and it is known as basement membrane. This can be observed even in light microscope. Or this basal lamella is fused with another layer, non-cellular layer. This is reticular lamella as observed in the deeper part. This together makes the basal lamella and reticular lamella together a thicker layer and become visible under light microscope. This is also known as basement membrane. So basement membrane comprises of two layers of basal lamella or a basal lamella along with the reticular lamella form the basement membrane. This is observed in the glomerulus of the kidney. You can see this rounded structure is the glomerulus and Bowman's capsule is the outer part that has a white line. Epithelium is outside that and this is showing basement membrane. Alveolar epithelium, this is alveolus, one sac is alveolus, and if we increase the power we can observe the epithelium are over the basement membrane. The functions of basement membrane are and the basal lamella are the same, and these are the selective barriers to the chemical substance or the fluid that wants to pass through. The second function is it guides the proliferation of the cell. When the cell divides, it spreads over it and helps the proper proliferation of the cell. The growth of the neuron processes is guided by this basement membrane and basal lamella of the Schwann cells and the supporting cells. This helps the neuron processes to grow and to reach the proper destination. Let us discuss the cell surface modifications. The specialized cell surface modifications are there to achieve some purpose. These specialized cell surface functions are to keep the surface of the cell clean or the area clean, and to increase the absorptive area in case of absorptive cell, and in some cases these specialized processes help the cell to move forward. Surface modifications may be microvillus, stereocilium, cilium, and flagellum. All are in singular number. Microvillus plural is microvilli, stereocilium is stereocilia, cilium is cilia, and flagellum is flagella in plural number. This is one figure. You can see in the figure the homogeneous area is lamina propria. This is connective tissue. Over there the arrow indicates the sheet of cells. So section it is showing as the row of cells. So row of cells are there. This is over the finger like projection, and inside this is lamina propria. So these are finger like projection of the mucosa. This is known as villus. So these are the finger-like projection on the surface of the mucosa. Let us take the cells from the surface. You will find that each cell has similar finger-like projection at their apex, and these are known as microvillus of the cell. These are so finger-like projections of the apical part of the cell, or it may be in other part. These are non-motile. They cannot move by itself. They can move only because of the flow of the fluid over it. Its a height is very small, and this is to see it very high power view is required. So it comprises of core of cytoplasm covered by plasma membrane. Let us discuss. This is on the left side. This is the electron microscopic photograph of four um, microvillus, and on the right side this is the diagram showing the structure. So each is comprising of a central core of cytoplasm covered by plasma membrane. This plasma membrane, lipid bilayer, proteins are there, carbohydrates are there. So inside the cytoplasm, we can see the network of cytoskeleton is there, working as the skeleton of the microvilla. So this one is relatively permanent structure. Two terminologies: brush border and striated border. This microvillas are these villi are so dense that they make a layer, continuous layer over the apical surface of the cells, and this is known as broad border and the striated border. The dense covering of the microvilli together is making a strip over the apex in staining condition. They are actually synonym, but especially in the small intestine we use the term brush border, and in case of the kidney tubules we use the striated border. The functions of the microvilli together increase the surface area, so the absorptive surface area is increased. In intestine we get it, this increases about 600 times area. In the kidney tubule, the microvilli increases the surface area for reabsorption of the absorbed fluid, filtrated fluid. Stereociliums are like microvilli, except they are very long than the microvillus. Each may be branched even. It is present in the epididymis. Epididymis is the part of male reproductive pathway. Ciliums are very, very long, elongated. It is finger-like, but the process is looking like a tail, very tall relatively. And these are having skeleton inside that helps it to move by itself. They are motile cell surface modifications. So about uh, 5 to 10 micrometer long, and in light microscope we can easily identify them with even lesser power. Each cilium has a very organized cytoskeleton. You can see the arrow is indicating the level of plasma membrane. So the cytoskeleton is inserted into the cytoplasm of the cell and having a very organized distribution as shown in the section. The microtubules are arranged in a circular manner, and they are well connected by other protein molecules. So this central core of cytoplasm is covered by the plasma membrane, and this cytoskeleton is very organized as I have described earlier. By their characteristics movement, the cilia can push the fluid or contain dust in the fluid on its exterior surface. You can see the one way they are withdrawing the cilium and making a thrush over the fluid. Here you can see the cilium are withdrawn. This is the return stroke. Here it faces minimum stress, and then they make a power stroke that with maximum stress to make a flow over the fluid over it. You can see this is the epithelium of the respiratory tract, and this is typically present in this epithelium over the apex. So lining epithelium of the respiratory tract is the best example of the cilia, and that's why it is known as ciliated epithelium. Flagellum. This is longest cell surface modification. It's a tail-like. One cell has a very long one process. It helps the cell to move forward. They help in swimming the cell or push the cell forward. This is observed in the spermatozoon, means sperm of the male. Basic difference between the four types of cell surface modifications. Let us compare each other. This is 4 columns are planned. Now one by one we will try to understand or compare these 4 types of cell surface modifications. This microvillus is the finger-like projection from the apical surface usually, and stereo they are relatively longer microvillus and may be branched. Cilium are very long. It's a longer surface projection than the microvilli. Flagellum is the very long. It is the longest type of the all. Microvillus is non-motile. Stereocilium is also non-motile. The cilium and flagellum are motile, but flagellum is very motile. Cytoskeleton form the skeleton, but it is simpler form. But relatively well organized cytoskeleton is observed in cilium as well as flagellum. The microvillus and stereocilium, they increase the absorptive surface area. The cilium keep the surface clean, and the flagellum helps the surface to move forward, give the cell motility. So the last row is the functional difference. Thank you very much for remaining with the presentation.